So I just have a few questions to ask you about the research opportunities that we have at URI, more specifically within the Menon lab. So can you briefly describe like the type of research that goes on in your lab specifically right now?
Sure. So in my lab, we work at the interface of two different kind of research areas, which is we have nanomedicine and we have tissue engineering, and we have projects in both of these areas basically. So in the nanomedicine area, we are focused on developing inhalable nanoparticle formulations that can be used to treat diseases in the lungs. So this could be lung cancer or lung fibrosis or COPD or things like that. And we develop multifunctional nanoparticles. So basically one formulation that can be used maybe to deliver two different kinds of therapies at different rates. So that's basically providing combination therapy, or you can incorporate an imaging agent so that you can image where these nanoparticles are going after they are administered. Or you can have a targeting component. So you can have these particles go and bind to specific cells so that the therapy is given only to the cell that we are trying to treat. It could be fibroblasts or epithelial cells or endothelial cells. So things like that.
Mm-hmm.
And then we have the tissue engineering component where we develop disease models in the lab. Because I'm sure everyone has seen people working with cells growing in petri dishes in the lab and using these cells to test different kinds of therapies. But you know that cells in the body are not present as just a flat two-dimensional layer. You see them as complex cell aggregates. So in the lab, we are trying to mimic those kinds of conditions, those cell arrangements, so that we can mimic how the disease is inside the body, so that when we then use these cell aggregates for testing different nanoparticles or different therapies, then the responses of these cells will be more accurate as well. Because in the last few years, when people have increasingly started to realize that when you grow cells as a flat layer in well plates or petri dishes or flasks, those cells, they don't respond the way cells in the body respond to therapies. So we are in dire need of better models in the lab. So we are focusing on developing these tissue engineered disease models for testing all these nanoparticles and other treatments as well.
What are some specific techniques or practices that you utilize in your lab, like the DLS and all that type of stuff?
Yes. So for the nanoparticle work, we use the DLS, dynamic light scattering instrument, which can be used for measuring particle sizes, how stable these particles are in solutions, how uniform their sizes are in the solutions, things like that. Then we have we use different techniques for lowering the sizes of these particles. We need to make them in the nanometer range, obviously, and it needs to be at least less than 200 nanometers. 100 nanometers is perfect, but at least less than 200 nanometers. So we use things like the sonicator, which can be used to break down our nanoparticle formulation so that they're really small. So we also use the confocal microscopy and EVOS images, which are basically used for seeing where these particles, we prepare fluorescent particles and we see where they end up inside the cell, whether they go to the nucleus, whether they stay in the cytoplasm, whether they're bound to the surface of the cells. That can be visualized with confocal microscopy. And then in the tissue engineering area, we have recently started working with 3D bioprinters. So we have two 3D printers in the lab that is being used for training the students in the classroom on different methods of developing tissue engineered organs. And so as an example, so I have this prop that I use quite often, which is of a 3D printed brain. And this is actually made with plastic. But we can, with the 3D bioprinters in the lab, we can develop this model using actual brain cells. And we can then culture like a three-dimensional brain tissue or in the lab, as I said before, for testing different kinds of therapies and, for modeling different kinds of diseases. And the other thing we have, and maybe you've seen this already, so we have this very futuristic looking couple of devices in the cell culture space. So we have this orb, which, I think somebody told me it looks a bit like an alien spaceship sort of thing. And what it does is it's used for culturing cells in different conditions within something called as organ chips. So this is an organ chip, and you can see that there are two channels. There's a pink channel and a blue channel in this, right? And what we can do is we can culture different cells in the different channels. So for example, you can grow endothelial cells mimicking the blood vessels in one channel. We can culture epithelial cells in another channel. And using this orb, we can mimic things like, for example, mechanical stresses. If you want to make a lung on a chip, you can introduce mechanical stresses, so you can expand and contract this chip. So the cells here are going to experience the same stresses that cells in the lung would experience during breathing. You can introduce fluid flow in these channels so that you can mimic how blood flows through the blood vessels, and what that flow and what that shear stress does to the cells, the endothelial cells in the blood vessels. So all of that can be mimicked with these organ chips. So and then we have technologies that we're developing in the lab. So we have microwell arrays, which are gels with little micrometer sized wells in which we can culture cells, and these cells will be in a three-dimensional format. And then we have scaffolds. We make porous polymer scaffolds on which cells can be cultured. So our aim with all of these technologies is basically finding the best method to mimic condition like disease conditions in the lab, whether it's introducing fluid flow, whether it's introducing stresses, whether it's introducing a substrate for cells to grow on. And if you're doing lung tissue engineering, then obviously the epithelial cells need to be exposed to air. So how do you do an air-liquid interface for these cells? So all of this is work that we're doing in the lab right now, and basically trying to find the best way to mimic all these diseases in the lab.
What would like a normal, I know it's very different depending on whose work you're considering, but a normal like day-to-day in the lab, or some of the requirements like our presentations, our lab meetings, and all that type of stuff. I don't know if it's different in other labs personally, but just like a normal experience, I guess.
That's a very good question. So when a student joins the lab, initially you have some lab trainings that you need to complete with the EH&S. So we'll schedule you for those trainings, and you need to pass them to then officially start working in the lab. And it's just you have some reading material that you have on Brightspace, and you will be able to say this more since you must have recently done this. So you would have some reading material, you'd have a quiz based on it, and once you pass the quiz, and it's a very easy quiz. If you've read the material, the questions are very straightforward. And once that's done, then you can start actually doing hands-on work in the lab. And initially, because some of the students coming in are very new, they have no experience being in a lab, I start off by having their, I assign a graduate mentor to them, and then they get to, or a senior undergraduate student. It's a graduate mentor or a senior undergraduate student mentor. And these students are asked to kind of start reading some papers. We send them publications on, whether it's nanoparticle work or tissue engineering work that they're doing, and be a little bit more familiar with the area in general. Like, why are we doing this research? Why is this important? And then slowly, the students start to, shadow the mentor, see the different techniques that's being done in the lab. And one thing I do to the students who come into the lab, and you know this as well, Nave, that I ask you to, whenever you have time, and you can come into the lab, and even if your mentor is not available, you can shadow a different mentor if they're doing something some research and you want to learn what it is, I always encourage students to do that. So if you have a mentor, it's not like you have to always be with your mentor. You can be with any senior student in the lab, shadowing them and learning all these different techniques. And then once you're comfortable with these techniques and you've watched it and you're comfortable making notes in the lab notebook, then you move on to actually doing hands-on work. Initially, you will do this, under the supervision of your mentor, and then once you're comfortable, you can start doing these techniques on your own. I ask students to present a paper during lab meetings if they don't have any data to show. But as the student, works more in the lab and starts to generate data, I encourage students to present that data in the lab meeting, because I think it's very crucial for you to be able to tell other people what is it that you do and why is it important, and you must be able to answer questions on your project. And so for that, it's important that you're able to present your data in front of your peers. Undergraduate students in the lab have also had the opportunity to present during research showcases at URI. And there are SURF students, so they present at the SURF program meeting. So these are all opportunities to talk about their work with other people and get used to answering questions. And it also helps you think more about your work and do additional studies based on the comments and questions you've received for your poster. So all of those are components that helps you develop as a researcher.
Yeah, it's definitely very intimidating if you don't know what to expect, because especially for myself, I didn't know anybody who did it, so it can definitely be a lot at first. But I mean, your lab specifically, like, I felt much more relaxed once I actually talked to everyone. You guys were like, don't worry, I don't expect you to know what you're doing. And I was like, oh, thank God, because I never worked in a research lab. And when I remember I walked in, I thought it was going to look like a teaching lab. And I was like, it's just one bench, but it's much different. So
yeah, it's very, very, very common for a new student to come in and feel like, do I fit in? What do I need to do? What do I need to know? But that's how everybody starts in the lab. The first semester is just a learning, you're just learning what's like in the lab, and it's completely okay to feel like, what am I doing here? But then I've seen very, very often that, by the second semester, everybody picks up and they're more comfortable doing things, because now they know the students in the lab, they know what needs to be done, and they're more comfortable.